Hello students, this is Dr. Amarpreet Kour, a lecturer of Jammu and Kashmir Education Department. Today I will discuss about the pistil, megasporangium and embryo. But before that, I request to all of you kindly keep the NCERT book of biology of class 12th open and open at the page number 24. Okay, there is a figure 2.7. It is labeled as a dissected flower of hibiscus showing pistil. Okay, and in bracket it is written as the other floral parts have been removed, so it's only the structure of pistil. And in B there is a multicarpellary syncorpus pistil of papaver, and diagram C is multicarpellary apocarpus gynoecium of Michelia. And the third one is a diagrammatic view of typical anotropous ovule. So I'm going to explain each and every term which is mentioned over here, and also I will discuss the pistil, megasporangium and embryo in detail. Firstly, with pistil. A typical pistil has three parts. One is stigma, other is style and ovary that you can see in the diagram figure 2.7a. Okay. Firstly, stigma. It serves as a landing platform for pollen grains after pollination. It is actually a receptor for pollen grains. Whatever are the pollen grains, okay, they come and they sit over this stigma. Then there is style. Style is it is a cylindrical tube, okay, and it actually attaches stigma to ovary. It is simply a canal that attaches a stigma and ovary. An ovary is the bulged part of a pistil, and it contains ovules. Before going ahead, I want to discuss some important terms like monocarpellary and multicarpellary. When a single carpel is present in a flower, it is known as monocarpellary, okay? And when multiple carpels are present, it is known as multicarpellary. Now, apocarpus and cyanocarpus. When carpels are free, means they are not attached with each other, then the condition is known as apocarpus. And syncarpus is when the carpels they are fused with each other. Okay. Now I am going to discuss about ovule. Ovule, it is integument indehiscent megasporangium. That means it is still attached with the parent plant and it is integumented. That means integuments they are still intact and it is not dehiscent yet. Kindly go to figure 2.7d, okay, for further details of an ovule. A typical ovule has 7 parts, okay, and they are known as funicle, first one, hilum, integument, micropyle, chalaza, nucleus, and embryo sac. The most important is embryo sac. Although all these have some functions, but in embryo sac is the future seed, or you can say the most important part from sexual point of view. First, funicle. It the stalk of the ovule by which it remains attached to placenta. See in the diagram, okay? It is clearly seen. It is a stalk of the ovule by which it is attached with placenta. Second is the hilum. The junction between ovule and funicle is known as hilum. Okay? Or you can say it is the point of attachment where where funicle it is attached with ovule. Now integument. These are the protective envelopes surrounding the ovules. Okay, not directly, but the body of ovule, and it can be one or many. Now micropyle. The pore or passage that is present at the tip of ovule where integument is absent. Clear? It is a pore or passage where integuments they are absent. Chalaza. It is the basal part of the ovule, okay, exactly opposite to micropyle. Now, nucellus. It is the parenchymatous mass of tissue that is enclosed within the integuments and it forms the body of ovule. Okay, it is simply mass of the parenchymatous tissue that forms the body of ovule. It is of two types. One is crassinucleate and the other one is tenunucleate. Okay, firstly the crassinucleate. This nucleate is well developed. The other one that is tenunucleate. This is poorly developed. Crassinucleate it is present in polypetally and in tenunucleate it is present in gammopetally. Polypetally it is the taxonomic class, okay, where the petals they are not attached with each other. Okay, while in gammopetally this is the taxonomic class where the petals they are attached with each other. Anyways, they were not considered in this period. They were only considered at the period of Carolus Linnaeus. Now embryo sac. It is also called the female gametophyte and it is located in the nucellus. An ovule generally has a single embryo sac. An ovule is of six types. Firstly, orthotropous. In this, the micropyle, chalaza, and funicle they all occur in a straight line. Anatropous. In this, the ovule turns at 180-degree angle. The third one is hemianatropous. The ovule turns at the 90-degree angle. That is why it is known as hemi. Hemi means half. Anatropous is it turns 180-degree angle, but hemianatropous it turns 90-degree angle. After that is compylotropous. The ovule is curved more or less at right angle to funicle. Micropillar and it is bent down slightly. This is the only difference. Otherwise, compylotropous and hemianatropous they are almost similar. The only difference is that the micropillar and it is bent down slightly. After that is amphitropous. In this, the embryo sac it is curved like a horseshoe. Okay. The final one is carcinotropous. The ovule turns more than 360-degree angle. Okay. So the funicle becomes coiled around the ovule. So carcinotropous ovule it turns to 360-degree angle and it becomes coiled around the ovule. Now all of you kindly refer to figure 2.8 okay of the same book at page number 226. It shows the parts of ovule that is showing a large megaspore mother cell, a dyad and a tetrad megaspore. Okay, see them. Now, megasporogenesis. The process of formation of megaspore from megaspore mother cell is known as megasporogenesis. This figure 2.8a, It shows the steps of megasporogenesis. The first diagram, the ovule generally differentiates a single megaspore mother cell in the micropillar region of the nucellus. Okay, at the micropillar region. This megaspore mother cell, which is present at the micropillar end, it has a dense cytoplasma and a prominent nucleus. Okay, this megaspore mother cell, it undergoes meiosis. That means it forms four haploid cells, or you can say four haploid megaspores. A tetrad is formed that you can see in the figure, out of which only three generates and one remains functional. This functional megaspore, okay, it is going to be the future embryo sac. Now, organization of embryo sac is very important. The arrangements of cells inside the embryo sac is very important from your examination point of view. So keep this detail in your mind. Open your book, go to figure 2.8. See, there you can see a mature embryo sac. At the micropillar end, there are two synergids and one egg. Now, what is the function of synergids? The function of synergid is, perhaps it is not yet confirmed. It is presumed that it secretes some chemotropic substance and it directs the pollen tube to grow inside embryo sac. Usually at the time of pollination, that means when pollens arrive to stigma, one synergid, it degenerates. In the figure, you can clearly see the filiform apparatus. This is special characterization of synergids. Now, egg. A typical egg has a micropillar vacuole and a clausal nucellus. Okay, there is a plasmodesmata connection. These are the connections for communication. They are present between egg and synergids. There are antipodal cells or vegetative cells. These are present at the clausal end. Usually there are three antipodal cells. The fourth one is central cell. It is the largest cell of embryo sac. Okay, and most important, it is 2n. It is diploid. Okay, every cell inside the embryo sac is haploid, but central cell, they are diploid. Okay, it is diploid. Sorry. Thank you for listening my lecture. In case you have any question, you can visit to my website, that is www.amarpreetkour.com, and you can post over there and you will find a number of lectures there. You can listen to them